Hello everybody, my name is Mehdi Kouri and this is another video about simulocyte. In this video we're gonna explore the editor and the layout editor for the models of the metabolic activity of the cell. So just to get back to what I was presenting before, we have seen before that we can build metabolic networks of the file, of the cell, and it's possible to edit layouts for this metabolic network. And once we have built this metabolic network, then we can start constructing a model of the molecular activity of the cell. And this model will take into account the metabolic network, connect to it, so that when we have a model with different genes being expressed and triggering, let's say, transitions that would produce certain things, these things themselves will be linked to metabolic activity, right? So I'm staying vague, and this is voluntary because I don't want to get too much into the specifics of the model itself. I just want to describe how you can edit it from a software point of view. Later there will be other videos dedicated to explaining what the model really is about and why it has been chosen as it is, and Andrei Kerzek will explain in further videos these particular specifics. Alright. So let's look at the models. Here we got a couple of models, 3 of them, and the first one is the old hepatocyte model. So let's just look at it. Let's click on show, and it's going to take, as usual, a little while to download all the information for this model. But what matters is we can see at the top there is like a QSSPN model editor. And then there's a new, there's a layout editor. So there's 2 different editors. One for the network properties of the model, which transition is linked to which place and so on, so as to form a network. And we can change what is linked to what. And then there is the layout, which is the purely visual aspect of the model. How do we want it to appear visually? So to get back to, from a pragmatic point of view, let's look at what we are having there. We got in blue our metabolic network, and we got in green our model. And this model has links in orange from nodes of the model, which seem to be linked to lots of nodes in the metabolic network. So as to give a general overview of the model itself visually, there's 2 circles. There's the first big circle there, which is actually places in the model. So these are round nodes. And there is another circle, which are the model transitions. So what are these? Generally speaking, the model is made of 2 types of nodes. Transitions, which are linked to places, and transitions have pre-places, like things which must be triggered for the transition to happen, or must be used as a trigger for the transition, or as an enhancer, or must perhaps prevent the transition from happening. And then post places, which are what the transition produces. Okay. If we look at what the model is, when we uploaded the model, and we uploaded 2 files. One is the QSSPN JSON file, which contain data about places, what are the types of places, state transition, and so on, and the transitions. And you can see that if we click on each transition, it has different tables associated to it, which gives different parameter, and this can be edited. I'm not gonna get exactly into the detail, but just to say that on this model page you can just edit the parameters, not the name of the places or transitions, because that would be changing the way places and transitions are connected, right? I mean, if you suddenly change the name of a transition, it's not connected anymore to the other places. So that's something that the editor will do for the network connections of the model. But here we can change the parameter value and save it by clicking on the update. So we click on a different transition, it has different activity tables and the list of what places are consumed or produced or so on. And we can just change. Let's say I click on that, there's an activity table with lower bound, upper bound, and all these things can be changed directly by changing what is written on the table and clicking on the update button. So this is the first way of editing the data. You also notice that you update not just the QSSPN JSON file, but you have to update to upload FBA file, so a file that contains flux balance analysis data. So if you want to edit, let's say, the structure of this green network, which node is linked to which node, you can go to the model editor and clicking here, and it's giving you a warning in this case because, well, when you make a model, every transition must have a different name, and in this case, this particular model it was an old model built by a team of biologists, and that model, unfortunately, they created different transition with the same name. So if that were to happen, you will receive a warning telling you that this transition's name, blah 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 blah, have the same name and they should have a unique name. And for the purpose of this editor, they are temporarily renamed with an extra number at the end to differentiate them. That means you won't be able to save any change you make in this. If you've got this mistake at the beginning, you need to upload a file with proper different transition names. But we're just looking at the editor from a user point of view at the moment. We're not really wanting to save the data. So let's just look at what's happening. How would you edit a complex network with transitions and places with perhaps hundreds and thousands of them quite quickly and quite efficiently? Well, it's actually possible with this kind of setting. Let me explain what's going on. There's two types of nodes. There's places and transitions, and let's look at a transition. I click on this transition here. Each transition has 4 different types of relationship with 4 different possible relationship with places. This transition can either be activated by a place or several places, can be suppressed by a place or several places, can produce a place or several places, or can consume a place or several places. And similarly, a place can activate a transition, can suppress a transition, can be produced by a transition, or can be consumed by a transition. And this is the heart of the editor there, because then it's possible to just select a transition in the list, all that being constructed by the file which was uploaded, and it's possible to just modify what this transition, for example, activates. Perhaps we want to add another place that this transition would activate. In that case, we just go to what is this transition activated by, and we just click another extra checkbox corresponding to the other thing. And it's possible to I'm just going to put it back to normal. It's possible to look at what also this transition is suppressed by. Perhaps we want this particular HNF4A transcription 1 is the name of this transition. We want it to be also suppressed by something else called LXRA, for example. This is totally random. So in that case, we do that, and we can then click afterward later on update save model data and proceed. You notice that there's another extra table here, which is what nodes in the metabolic network are linked to whatever you have here. So generally these are places which are linked to the metabolic network. So if I try to find a place in the lot which is linked to metabolic network node, let's say perhaps something with the word cholesterol, cholesterol B, what's that? Yeah, well, it seems that cholesterol is linked, cholesterol B for example. Yes, good. Cholesterol B is a place which is linked to a particular metabolic node, and it's possible to add different extra metabolic node to say that it can regulate other metabolic functions. So yes, you can just very quickly, intuitively select anything and add what you want it to activate, suppress, produce, or be consumed by, and so on. So this is very, very fast because you can do it for one particular thing, but you could do it for several. So let's say I want to create several things like this. All this, I want all of this to be to suppress one particular new transition. So I select all of these places, and I want all of this place to have this extra suppression to this particular degradation, and that's it. It's done. So we could select 20 places at the same time, or all of them, and decide that they're all produced by a particular transition. Or to make it more interesting, perhaps we can select a transition or a group of transition all at the same time, and just decide that they all consume ATP. So that's a way to create, edit a network in the model, edit the model very quickly, and suddenly create perhaps 200 different transitions that all consume this particular thing called ATP. Also, once you have edited your model and created new things, for example, this particular transition might consume some other thing called MATP, well, why not? Then you can update the content of the rules that result from your model. And these rules, then, once you select them, you can visualize them as a graph. So if you select the whole thing, you can visualize your whole model. Or if you select anything that contains a sub-portion of your model, like then you can, let's say, degradation. And then suddenly you can select anything that contains the word degradation in your rules, which are derived from the model that you've just edited. And suddenly you get that particular big graph. Perhaps you should change the layout to concentric. And then yes, you can start seeing, the network of your model itself, when the sub-portion of your model, which is all about degradation. So yes, you click on update, save your model data once you have edited your table, and proceed. And then, unless you've got an error message that tells you that some transition have the same name, then you'll be able to save the change. If you have transitions with the same name, then it's going to tell you won't be able to save the change, of course, so as to prevent errors to propagate when you build your model. So that was the editor for the model. It's a very, very fast way to edit and create models. You can also add new transitions or new places, because at the end of the table you would just enter a new name and click on whatever it would suppress or produce or whatever. Yes, so I'll get back to the models, and I'll now tell you a little bit more about the layout editor, because we have seen the model editor. We can edit the model, the parameters, the way the nodes, the transitions are linked to which places, and so on. But we can also edit the layout the way it visually appears. So let's say I want to add a new layout. I want to make it available to everybody to read, so and I want to call it hep layout model. I have model layout video 1. Okay, and I want to make sure that it's linked to the old hepatocyte QSSPN model, which is this one I created. And then I'm going to get to the editor screen. So it's giving me the same warning as before. Some transitions have the same name, but that doesn't really matter because we can save the layout anyway. It doesn't care about the content of the model. So yes, we got that. What we do is we can just select a particular word or a particular portion of your model that we want to visualize, or the whole model even. Let's say, you know, if with the mouse I select one column, you'll see the associated graph, the rules that correspond to the graph. So if I select all the rules, then I'll have a giant graph, but perhaps I'm interested in giving a particular layout to a sub portion of the graph. It would be like a subgraph being differentiated from the whole rest. So anything which is about, if not degradation, perhaps hydroxylase. What is that? I'm not a biologist, but let's just anything with the word hydroxylase. Okay, anything with the word hydrox will be isolated as a different kind of subgraph. So I'm going to create perhaps a concentric layout, yeah, so that there will be a subgraph. It's possible to select everything or a subcomponent, drag, drop, whatever. It's possible to grab one individual node, pull it, and you know, give it a different layout. Perhaps yes, let's make it, let's see, what is the grid? Now grid is not very good. The default layout, it's too far away. Circular layout is not very good. Concentric is actually the clearest layout in this case. So I want to have a concentric layout for everything linked to hydroxylase in my model. So it would appear as a subunit. So I'm going to save it as hydroxylase. Hydroxylase. Yes. And I'm going to save that layout. And I'm going to proceed. And then once I have done that, it's going to redirect me to the QSSPN model, and it's going to be possible to now select that new layout that I've just created. And it's possible to, for one model, create lots of little subparts with different layouts. So you can, you know, let me choose for the layout. It's called hep model layout video 1. Yeah, that's this one. I select that layout, so it's going to refresh the whole data and give me a point-by-point network layout of all the subpart that I've done. This could become a big layout, quite big from a data size point of view. That's why I have to refresh things so it just grabs the right thing. So yes, so I've chosen this layout. So now what subgraphs are available? Well, there is something called hydroxylase that I've just created. Yeah. So let's first, the whole model is placed in a little bit badly. Let's put it to the left. Yeah. So I click there, and I put it to the left. And now I want to look at this subgraph called hydroxylase I've just created, and then hop, it's just going to put my hydroxylase. And remember, it has this kind of circular pattern with a circle at the center and a circle outside, and you can see it's now in 3D, so they are kind of above each other, this circle, but I want to reduce their separation. So I'm gonna click on the separation thing to make them just on the same plan. And you can see this corresponds to exactly the same layout as the one I defined earlier, and it's linked to the rest of the model and also to the metabolic and the metabolic network, and we can also displace it to the left and the right, because that's what I've selected so far. So let's say I want it to be a bit more on the left. Yes, and that's a nice way. If I've got other subcomponents in my model, I can visualize them apart, and I can literally deconstruct the whole model, which has this default big circle confusing thing, into sub-part connected, and I save them by keyword, and that's very, very convenient as a visualization tool. It's possible to place them in 3D up or down. Let's say I want actually that thing to go, at the bottom for some reason. And yes, and that's it. I've got the very nice layout for my model. So that's it so far for this video. I've shown you how you can edit the model, the parameters in the model, which are in the main screen itself here on the table, but also the connectivity of the model in the model editor, and I've shown you how you can create and edit a visual layout of your model. Next time we'll have a look at how to create experiments, and we'll also have a look at, once an experiment is created and ran, we get simulation data about what happens to the cell, and we'll see the detail of what kind of shiny things can be visualized in 3D regarding flows and sequences and all sorts of things that happen to this virtual cell. Alright, thank you very much for listening, and bye bye.